Okay, good morning, ladies and gentlemen. Time here to start today's lecture. Thank you. So first thing to say is we're quite a tight fit in G2. I was working out. So whenever you come in, if you actually all sort of fill to the edges of the seats, then it's probably best. And we also got a dramatically smaller screen in G2 as well, but hopefully everybody will be able to read everything on there without too much trouble. Right, so this is MBB 340 or MBB 6340 if you're one of the MSc students. The microbiology of stream environments, again, a bit like the cells as factories turned out to be this year because Dr. Partridge was on strike for her lectures. There's just two of us teaching in this module. I have the lion's share of the lectures, and then Dr. Baker comes and talks specifically about protein structure for halophiles, psychrophiles and thermophiles. So the lectures really should hopefully dovetail really quite closely between myself and Dr. Baker. So these are the lectures. There's obviously one today and tomorrow. There's no lecture this Friday because I can't be in two places at the one time. I've tried it before, it doesn't work. So there's no lecture on Friday of this week, and there's also no lecture on the final Friday of the module, the 20th of March. So in total there's 16 lectures, 12 from me and 4 from Dr. Baker. Of course, the student handbook on the MOL page. My lecture slides will go on there, hopefully at least the night before. If I'm better that, I will do lecture 2 is already on there, for example, for tomorrow. And those of you who came to MBB 303 or 6303 sort of know the way I organise things. I'm trying to set up a framework of the course for you. I do expect you to do the reading that I do sort of draw your attention to. And indeed, I do put the papers, PDFs of the papers, onto the MOL or the Blackboard page, as it should be now. And to make it as easy as possible to do some of this extra reading so you can incorporate that into the essays in May or June. Okay, so that's by way of introduction. I'll have 16 lectures, 12 from me, 4 from Dr. Baker, and these 2 Fridays there's no 303, 6303 lecture. So I'm not going to show you all of the student handbook. You can go and have a look at that for yourself. But I just thought I'd just show the learning outcomes part of it. There's 3 learning outcomes. The first one is essentially me. So I'm going to talk to you about a whole bunch of extreme environments, and I'm going to tell you, or hopefully explain, how organisms live in these extreme environments. And one thing that I'll make clear today, but it comes into my head straight away as soon as I look at this is optimal growth in these extreme environments. This is not things surviving. I mean, it's interesting something can survive in a very sort of high temperature or low temperature. These are things that are growing there, and sometimes growing there quite quickly. And as I say, Dr. Baker will come along and look at the proteins, how they're an integral part of the way the organisms can grow in these environments. And then, obviously, we want you to appraise and think about the techniques and results that we're presenting to you, so you have an overall feel for this area of extreme environments and the microbes that are found there. Okay, so let me first of all define an extreme environment. And this one on the slide just now is what I've called, a very big word, an anthropocentric, I can't even say it either as well, anthropocentric definition. So what that means is that's from our point of view. So obviously, if I took you to a hot spring and pushed you in, you would not be very happy with me, because at 90 degrees centigrade, you get very badly burned. Equally, the pH might be quite acidic as well, so not only you'd have temperature, you'd have also problems with pH. So clearly this is not an environment we'd be happy in. We could wear some sort of hazard suit and go in some ways, but that's not the point. The point is that human, under normal conditions, would not be happy in many of these extreme environments. It'd be too hot, too cold, too acid, even too alkaline for human life. So you could sort of define, and it gives you an idea of what sort of environments we're talking about, but really a better definition, in a really a simpler definition, is an environment where the species diversity is very low. So you don't find lots of different species, and almost all of the species you do find are microorganisms, or in some cases very simple invertebrate organisms may actually survive. But the vast majority of the really extreme habitats I'm going to talk about are almost totally dominated by microorganisms. So that's a more scientific definition. It's telling us that these are environments that are very selective for the organisms that can actually survive in the environment. So I think the first one, the anthropocentric one, is useful for setting a sort of feel for these are extremes in terms of what we would think of as extremes, but really the scientific definition is a better one in terms of how would we actually look upon an extreme environment from a scientific point of view, and we'd actually think that it was an environment that was very low in species diversity. A point I'm sure I make later in the slides, but worth mentioning here, although the species diversity is low, the biomass may be quite high. So it doesn't mean to say that a salt lake, for example, has got no life in it. A salt lake has got lots of life in it, but it's actually very restricted to halophilic bacteria and some other organisms. So they can be very rich, these environments, in terms of the amount of organic matter produced by living organisms, but the diversity of the organisms is really quite low. Okay, so what about types of extreme environment? So the first one I want to mention is where you've got low water potential. So low water potential means you've got lots of solutes in the water, often salts like sodium chloride and other salts. But in principle, a low water potential area could also be a sugar solution, for example, not a very natural environment, but one you could find in an industrial setting. So again, in this case, there's still a lot of solute in the water, making it a very high osmotic pressure, very, very low water potential. So if water potential is low, osmotic pressure is high. And this can be things like salt lakes. I'll show you some examples of them later in the lecture. Or it can also be salted food. So a way, not so common these days, but it used to be very common in the past of preserving food, particularly fish, was to actually salt the fish. And some of the fish would become contaminated with a red organism, and this turned out to be a halobacterium. So they will actually grow quite nicely in salted food as well. The next one then are thermophiles. So these are the ones that grow in very high temperatures. So hot springs, which you find mainly in areas of volcanic activity, either on the surface of the Earth or under the deep ocean, the hydrothermal vents that I talked about back in first year. And like the salt lakes, these are quite niche environments. So what I mean by that is there's not much of the biosphere that is made up of either salt lakes or hot springs. So a very small percent of the whole biosphere is suitable for these extreme thermophiles and extreme halophiles. But where they are found in quite large amounts, the organisms themselves. Now I make that point because the next one, the low temperature, so the other side of the temperature coin, this is not a niche environment. A lot of the Earth is low temperature at the end of the day. All the polar regions, and also most importantly, the bulk of the oceans. Once you go below a certain depth, even in the tropical parts of the globe, temperatures fall very quickly, and deep ocean is very sort of constant, 4 degrees centigrade or so. So you actually have got a very large potential environment for these psychrophiles to grow in. So that's to contrast that as a big habitat, if you like, against two much smaller habitats. So the fourth one then is acidic habitats with low pH. Certainly there are natural acidic habitats, but a lot of them that we commonly look at in the 21st century tend to be caused by human activity, for example mining for various elements. It often leads to a runoff coming out of the mine that is really quite acidic, and also rich in metals such as iron, and also rich in sulfur. And again, these acidic habitats are fairly niche. Again, there's not many places on the Earth where you've got extremely acidic habitats, but they are reasonably sort of spread out all around the world. The nearest one probably to here, you could get some in Derbyshire as near as that, but certainly on Anglesey, the island off the northwest coast of Wales, Paris Mountain, there's actually there's a mine there with very nice acidic habitats that you can actually study. So we don't have to go too far to find a nice acidic habitat. And then finally, or almost finally, alkaline areas. And again, we flip the coin again. There's lots of parts of the Earth that are alkaline. Deserts are all alkaline. Soda lakes are the classic alkaline type of environment. But many soils are actually quite alkaline. So depending exactly how you define how alkaline it has to be, quite a bit of the Earth can come under the heading of being alkaline. Okay, so we've got halophiles, then we've got thermophiles and psychrophiles, two sides of the temperature coin. Then we've got acidophiles and alkaphiles, two sides of the pH sort of range. And the final one that I'll mention are the high atmospheric pressure organisms, now called pisophiles. They were formerly called barophiles, so if you see this word barophile, then just take that essentially as a synonym, meaning the same thing as pisophiles. Some people do try to make a slight difference between the two, but I think that's been lost. I think now it's really older literature calls them barophiles, newer literature pisophiles. And these are organisms that grow very well at hundreds of atmospheres of atmospheric pressure. So the hydrothermal vents I mentioned a few moments ago, the organisms there have to be pisophiles, because every 10 meters you go down to the ocean, you essentially increase 1 atmosphere. So at 5 kilometers down, you've got 500 times atmospheric pressure. And so we'll start to say a little bit about the pisophiles towards the end of my lectures. So that's the 6 groupings then that I will cover. Not all in exactly the same detail, but hopefully I'll give you enough detail on all of them to give some insight as to how organisms grow in these habitats. And one thing to point out is that you can, of course, have more than one stress factor. So the one I just mentioned, pisophiles and hydrothermal vents, well, of course, you're going to grow near a hydrothermal vent, you're almost certainly going to be thermophilic as well. So a thermophilic pisophile is one combination. You can actually have, sort of cycrotrophs that are pisophiles if they're growing in the depth of the ocean, but not near a hydrothermal vent, and so on and so forth. And I'll say something about these polyextremophiles again in a few lectures' time. Even at this early stage, if you ask me to nominate what is the most extreme of all the extremophiles, the thermoacidophiles must be pretty close. These are the organisms that grow at 100 degrees centigrade and pH 1, i.e. boiling acid. I think that's pretty impressive by any standard. And that's also why I have a very strong feeling or hunch, or what you want to call it, that there certainly are microbes elsewhere in the solar system. When you look at the Earth and where things can grow on the Earth, some of the other places in the solar system, the planets of Jupiter, things like Europa, definitely have conditions that are not so far away from things we can see on Earth. And it would be very strange if there was not at least some life there. And I'm hoping that we manage to get, we being the human race in this case, manage to get some probes to some of these really interesting places and actually show whether there actually is microbial life there, and crucially, is it microbial life based on DNA, RNA, protein, etc. So hopefully that can happen sometime in the next couple of decades, and we'll find out whether there is at least other life in the solar system. And quite a bit of the funding for extreme environments and this sort of work now comes from people like NASA, the space agency in the US, because they've become very interested in this idea of extremophiles and where they might find them elsewhere in the solar system. Okay, so just a couple of pretty pictures for this first lecture. So this is the Dead Sea between Israel and Jordan in the Middle East. This is a very highly salty environment, and as I'll tell you in a little while, it's not similar to seawater. So some salt lakes are concentrated seawater, i.e. they've got sodium, chloride, etc. in that ratio. The Dead Sea is very rich in magnesium. due to the underlying geology of this area. So it's a very strange place, the Dead Sea. You can literally lie on your back and read a newspaper and float on it. It's very sort of viscous. They have lots of showers, simple showers set on the shoreline, because once you come out of the Dead Sea, you have to wash off the salt, because the salt, if it crosses onto your skin, would actually be very painful. But on the other hand, many people go to the Dead Sea because they have got problems with skin, psoriasis, eczema, and it's felt that the sort of bathing in the Dead Sea can be beneficial to some of these skin problems. So it's certainly a very interesting part of the world, and a place I've never yet managed to get to. It's been on my list of places to go to for the last X years, but hopefully I'll get there eventually. One place I have been to is the Great Salt Lake, just outside Salt Lake City in Utah in the USA. And this is concentrated seawater. So this lake has got the same ratio of sodium chloride, potassium, sulfate, so on, as seawater, but it's just very concentrated. And you do get some things living here that are not microbes. There are a few brine shrimps that grow live in the Great Salt Lake. But again, it's massively dominated by microorganisms. This sort of area in the foreground, that sort of greenish, yellowish stuff, is actually muddy sulfur stuff. I've stepped into that and almost sunk down to my knees. And to my eternal shame, we actually left these Wellington boots in the hotel room we're staying in for some poor person to come along and open a plastic bag and find these things stinking of sulfur and other horrible things. But I just couldn't find anywhere else to get rid of them. So it's often the case in extreme environments. They can be a little bit tricky to explore, and always be careful when you're doing so. And I think the next one picture, I think there's one more. Yeah, there's one more salty environment. So this is actually San Francisco Bay, but it could be, for example, Malta in the Mediterranean. Many other sort of shore communities do this sort of this type of thing. And this is actually an industrial facility in the US to actually make salt. So essentially they're taking in water from the bay and allowing it to evaporate through a series of ponds to eventually end up with the salt. The red colour is due to the microorganisms, mainly Halobacterium, but some Dunaliella as well, which grow in the initial ponds until even for them it becomes eventually too salty. So the final ponds are white with only the salt. But this is an age-old process, as I mentioned Malta, because there's some very ancient salt pans that probably date back to the Roman Empire times, 2,000 years ago, when they were actually again producing salt in this fashion. But again, you can see the microbes are having a big effect on this facility. And this was the other health and safety one that I just mentioned a moment ago. This is Yellowstone National Park. This is a hot spring, a sulfur hot spring. You see it's very yellow. This hot spring is about pH 1 and 2. It's probably 70, 80 degrees centigrade. And this guy is completely crazy, in my personal opinion. I would not sit where he's sitting, even though the other guy is kind of roughly held around him. Apart from one thing, once you sit down in that sort of stuff, once you take these trousers off and wash them, they'll come out of the washing machine as just one big huge hole, because that's essentially sulfuric acid that he's sitting on to all intents and purposes. Then anyway, for his science, he wants to get a sample, and he's also using a long sort of pole and net to get the sample. Again, all kidding aside, Yellowstone National Park is a beautiful place. I really recommend if you ever get a chance to go to it. But you always have to follow where you can actually walk. So again, it is possible to walk near these and actually step through the crust into one of these sort of hot springs. So you have to know what you're doing when you go to sample them. But again, a very interesting extreme environment. And finally, this little sort of set of pictures. This is much closer to home. This is a massive tar lagoon somewhere in northeast England. Of course, if you come from anywhere near where Hoole Bank is, you'll obviously know where this somewhere is, but it's supposed to not be, I'm supposed to not tell people where it is. And bottom left, that's what it looks like. It looks like a nice little pond or small lake. But what it actually is a pit, a clay pit that's been excavated for clay. And then in the 1960s, the chemical company is based in Runcorn, Merseyside, up along the Mersey from Liverpool, were allowed to actually dump lots of chemical waste here. It was sort of semi-legal, and this is actually now full of all sorts of horrible contamination. You can see in the bottom right, taking samples from it. In the top right, Leanne Sunder, who was the student who did this work about 10 years ago now. And what we're looking for was any organism that could actually help to bioremediate this acid tar lagoon. And we found a number of organisms that would grow at these very low pHs in the presence of all this sort of organic material. And you see we had to wear the white, hazard suits because I say the problem is that nobody really knows what's in this lake. That's the problem. There could be literally anything. And certainly a lot of organic compounds like benzenes and things such as that. So some of the salt lakes, some of the salt lakes are high pH, so these are soda lakes, and they produce nice pink sort of haloalkophilic bacteria. And you get these lovely pink flamingos, which people like David Attenborough love to go to, the Rift Valley in Kenya and show you pictures of. It doesn't always tell you, of course, all the pink colour comes from the halobacterium. It's not made by the flamingos at all. It's all due to the fact that they're eating these bacteria. With the carotenoid in there, it makes this lovely sort of colour for the organism. Now I think this really is final this time. Yep, deep ocean hydrothermal vent. I talked about this a couple of in first year. These idea of these vents at the bottom of the deep ocean. This is one of the white smoker ones. Just a picture from Wikipedia, just showing you superheated steam coming out of the cracks in the bottom of the ocean. And you get organisms growing here once the temperature cools down a bit to allow even thermophiles will not grow much above 120 degrees centigrade. All right, so that's just by way of a sort of general introduction, because we're in the first lecture of the course, showing you hopefully some nice sort of pretty pictures of things. So I'll just continue a little bit more introduction, and I'll start with the halophiles and just see how far we get to. Okay, so just give you a bit more detail on the biodiversity in extreme environments. I've already said there are very few multicellular organisms. There's essentially no animals or higher plants. Probably the most complicated thing you might get is something like a brine shrimp, an invertebrate organism. But they're totally dominated by microbes, and even within the microbial world, they tend to be dominated by the prokaryotic microbes, both the bacteria and the archaea. But we will talk about eukarya as we go along the way, particularly when we talk about psychrophiles and halophiles. There are quite a number of algae and fungi that are both psychrophiles and halophiles. But a lot of the high temperature environments, for example, and the highly acidic environments are very dominated by bacteria and archaea. And we have the problem that we have with all sort of bioprospecting and looking at the biodiversity of microbial life, that we always underestimate it if we do it by some sort of growth technique. So if you go to a hot spring like the guy was sampling from, and you then plate out these organisms in some way, or you put them into liquid medium, you'll get some organisms growing, but probably only a few. If you then go back to the sample, or take another sample, and measure the amount of sort of ribosomal RNA, 16S in particular, for these prokaryotic organisms, you'll find lots of distinct 16S sequences, far more than you found organisms that will actually grow. So clearly, this is a big problem in sort of environmental microbiology. The idea we know lots of things are there, but when we try to get them into pure culture, then we actually lose a lot of the diversity. For example, in that acid tar lagoon that Leanne worked on, we isolated 6 organisms, by sort of enrichment in broth cultures and plates. But when Leanne did the sort of molecular biology side of things, we reckon there are at least 60 or 70 different microbes in the acid tar lagoon. The good thing about extreme environments is they tend to be quite low levels of, relatively low levels of species diversity. As I said right from the beginning today, if you go to an agricultural field and take 1 gram of soil from a farmer's field, you'll be overwhelmed by literally thousands, tens of thousands, hundreds of thousands of organisms, quite literally. So at least in an extreme environment, you get some you can just about handle. So something like 60, 70 microbes that are fairly dominant in the acetyl lagoon, but again we could only actually find 6 of them on plates. So we know we're missing quite a lot of things, and it's an ongoing sort of challenge to try and work out ways of isolating the other organisms. It may well be that they're living as consortia, so that organism A requires organism B to do something before it can grow. So if you try to grow organism A, even in a lovely medium, it's looking for organism B and it's not there. So growing things as consortia may be one way forward, but then it's much more difficult to work out the properties of organisms if they're not in pure culture. So it's an interesting sort of business to try and overcome. Now I think extremophiles are important anyway, but if you're writing a grant proposal or sort of saying why should you come to this lecture course rather than the one down in F2 at the moment, then we can say things that extremophiles are important for various reasons. So one is what we essentially covered in MEB 303 before Christmas, the idea that you can get lots of valuable products. In that case, I was emphasizing the algal world at that time. So I'll say a little bit about biotechnology again in these lectures, looking particularly at bacterial extremophiles more than the algae. But these are sort of a list of things that you can get from extremophiles: antioxidants, biostabilizers, the compatible solutes. Again, if you did 303, we talked about that before, and biosurfactants. So surfactants are essentially detergents. If you have anything, if you've been cleaning your room this weekend or whatever, if you've been spraying something onto a shower or whatever, or surface in the kitchen, this will have a surfactant in it. And there's a big move among many companies to make these surfactants biosurfactants, which will break down more easily and be more green. So this is a potentially big market for for people who can actually produce detergents from natural organisms. They're also very interested in the study of biodiversity because they give us an insight into how some parts of the evolutionary tree have developed. Interesting enough, if you take something, say thermophiles, they're not found only in one group, they're not even found only in one domain, so they're spread quite a long way throughout the microbial tree of life. And so there's quite a lot of interesting sort of insights you can get into the phylogenetics by looking at extremophiles. And as I mentioned a few minutes ago, because the diversity is lower, you can do things with metagenomics and look at a whole sort of salt lake biome much easier than you could a more complex sort of area. So I think for biodiversity they are a very good way forward. And they're important in understanding the origin of life, because the hydrothermal vent theory of life is certainly my favourite one at the moment, and that clearly would have meant that the first cellular organisms were most certainly extremophiles. It's likely that life evolved in a fairly hot Earth one way or another, so you can certainly think that thermophiles go back probably to the origin of life. So some understanding of what the original organisms might have looked like you can get from some of the very sort of simple thermophilic organisms that when you do the phylogenetic tree you really come quite close to the universal ancestor. So again we can get insights into the evolution of life on Earth and the origin of life by looking at a whole range of extremophiles, but particularly thermophiles. And finally, as I sort of mentioned in my introduction, they are really important for investigating extraterrestrial life. So the idea that we could have found something that grows at pH 100 degrees, then means that we can find similar sort of habitats elsewhere in the solar system, then it gives a real possibility of finding life. The key thing is you have to have water. One thing that is certainly true, and I'm always very reluctant to make any general statements about biology, because there's always an organism somewhere that waves its flagella and said, no, I don't do that. But I think it is definitely true that you do need liquid water for life. I can't think of any example where something will grow actively if there's no water present. So anywhere where there's water, then that would be great. If you're interested in science fiction, people have hypothesized, sort of gas planets, with no water having life based on gases. It's entirely plausible, but we have no scientific insight into that. But it would be fantastic if that was ever found to be true. Okay, so that's a sort of introduction to my whole part of the module, really. I'm going to go through these groups of extremophiles. I'm going to tell you a bit about them. There will be a little bit of overlap with MBD 303, but nothing dramatic, and also I'm aware that many students are doing 340 on its own, and that's absolutely fine. If there's anything you really need to know, I'll make sure that I tell you in these slides, so there's not any need to have any revision or try to find any slides in the other module. Everything will be self-contained. I'll explain mechanisms as far as I can, but I'll certainly come at times to a point where I stop, and basically Dr. Baker will take over that sort of argument about to do with some of the protein structure in particular, because he's far more expert at understanding proteins than I am. And then we'll do a bit of polyextremophiles, the ones that have got more than one stress that they're happy to grow in, and then find a little bit of biotechnology at the end, but not a huge amount. This is more a sort of basic, more general sort of microbiology module. Right, so I'm going to start with halophiles for the simple reason that I'm biased towards halophiles. These are the organisms I've studied most myself and know most about. So you get a little bit more in halophiles than one or two of the other ones, and that is entirely due to me being biased towards them. Okay, so where do we find halophiles? I've alluded to this a little bit already, but I'll give you some more detail now. So we find them in inland lakes, like the Dead Sea, like the Great Salt Lake. Many of them are in tropical areas, and essentially the evaporation of water exceeds the input, so the lakes become more concentrated and fairly stably high salt. Normally the temperature is going to be high, but the pH can be quite variable. So some salt lakes can be sort of highly alkaline. These are the soda lakes we'll talk about later on. Some are fairly neutral, and some can be a little bit acidic. It really depends on the geology of the rocks surrounding the lake. So, for example, in the case of the Dead Sea, the River Jordan is the only input of fresh water into the Dead Sea. And the Dead Sea is actually shrinking quite dramatically. And the reason it's shrinking is because water has been abstracted from the River Jordan for drinking water and irrigation by both the countries of Jordan and Israel. You may not be too surprised to find out that they don't agree always on the quotas of the water to take. And so the Dead Sea is literally shrinking. And it's quite worrying, in fact, because the Dead Sea could literally entirely disappear. But hopefully they'll find some way of preserving it before that happens. The other thing I showed you in the picture is salt. Well, actually, they're called salterns. I don't think I actually mentioned that name. So these evaporation ponds for salt production are called salterns. And these are made for industrial purposes to make salt. A lot of the salt made in this way is not refined, just used for rock salt for de-icing roads and things like that, but it can be purified into table salt if necessary. So salterns. And then you get microbial mats sitting on top of lakes. These are often dominated by cyanobacteria in this particular case. So rather than being planktonic organisms, sort of swimming or floating in the bulk of the salt water, they grow as a sort of crust on the surface of the salt lake. And then the one salty environment that we really have in the UK is underground saline deposits. So you can actually obviously mine salt from in the way you mine coal and other things. There are a number of big salt mines, obviously not surprisingly in Europe as well as the UK. And scientists have gone down into these salt mines, very carefully sort of taken a salt crystal from a very newly sort of exposed part of the mine, taking it back to the surface without letting the inside of the salt crystal come into contact with anything, and then isolated halobacteria from the center of the salt crystal. And they can then work out that that salt crystal has been in that position for maybe 200 million years. So the claim is that some organisms have actually survived in salt for this length of time. It's always controversial because you can never totally rule out the possibility of contamination. But to be fair, what's the chance of getting contamination of an extreme halophile if you do work quite carefully in a clean environment? So I suggest that it's fairly robust work, but still controversial. But people are very interested in the idea that these halophiles have been there for not thousands, but literally millions of years. So how do we classify them? Into a sort of continuum, essentially. You can have halotolerant ones, so these are the bulk of microorganisms that don't require any sodium chloride, or if they do, it's only literally millimolar. But they can range up to maybe as much as 1 molar sodium chloride. And just with these malarities in sort of some sort of context for you, seawater has an equivalent osmotic value of about 0.5 molar sodium chloride. So something like 1 molar sodium chloride would be twice the total salinity of seawater. And taking E. coli growing in very rich LB medium with everything it wants to grow, will grow up to about 0.6, 0.8 molar sodium chloride, not particularly happily, as they said, but they will just about grow a bit at that level, but they grow massively much better at low salt concentrations, so they're clearly a halotolerant organism. We then have a group of moderate halophiles, which have got an optimum growth between 0.5 and 2.5 molar sodium chloride. So what this means is that they require seawater levels of salt to grow optimally, and not surprisingly, most of these organisms are seawater bacteria. You find them mainly in the sea, but they can actually range up even as high as 4.5 molar sodium chloride, some of them. But the key difference between a moderate halophile and a halotolerant organism is you can clearly demonstrate that a moderate halophile requires a significant amount in the region of 0.5 molar salt for optimum growth, whereas with the halotolerant ones, they require almost no or very little salt. And then we have the ones that you find in the Salterns and the Great Salt Lake and the Dead Sea, the extreme halophiles, and their optimum growth is above 3 molar sodium chloride, so we're getting up to 6 times equivalent seawater salinity, and they won't grow at all below 1.5 molar. So you need to give them 3 times salt salinity of seawater for them to grow at all. And they will grow all the way up to saturated salt, which is about, well, pure sodium chloride, it's about 5.2 molar. If it's mixed with other salts, as it would be in a natural environment, it would be a bit less than that. So you ever see somebody sort of presenting a poster at a conference and they say they've grown their organism 7 molar salt? I'm afraid they haven't, because you can't get any more than 5.2 molar salt into solution. So just going back to the Dead Sea for a moment, quite a few years ago now, we had a bright idea in my lab. We were going to actually grow some organisms under exactly the same conditions as in the Dead Sea. So we got all the most latest measurements from the Dead Sea from colleagues in the university in Israel. And they gave us all the things, all the amounts of salts that were present. We made this up absolutely carefully, and it wouldn't dissolve. So we just could not get the Dead Sea to dissolve in the lab. And it's not really that surprising. I mean, it is obviously an amount of salt or mixture of salt that is just on the edge of coming out of solution. So that was a really interesting summer project. We tried everything we could to try and get exactly Dead Sea water, and we just couldn't get a Dead Sea medium that was exactly the same as the Dead Sea. Right. So We know there are these niche habitats, these salt lakes around the world. We know microorganisms are growing in there. We can go to them. We can look at them under the microscope. We can get quite a few of them into pure culture. But how do they do it? How do they grow in 3, 4, even 5 molar sodium chloride? So what are the mechanisms of salt tolerance? So the first thing to say in this section is, and again, luckily it's another general statement I can get away with this morning, I think, is that no halophile is the same internal sodium chloride concentration as the external concentration. So if somebody's growing 4 molar sodium chloride, it does not have 4 molar sodium chloride inside the cell. But it must have an osmotically equivalent amount of something osmotically active within the cell, either salt or other compounds, to maintain the osmotic balance. If it doesn't maintain the osmotic balance being a single cell, and these cells we're talking about don't have anything like contractile vacuole or some eukaryotic organisms of ways of pumping water out of the cell and things like that. These bacteria have nothing like that. If they don't match the osmotic potential outside the cell, they will lose water and become dehydrated. So although they don't use exactly sodium chloride, they must use something else. And what it comes down to is that there essentially is 2 main mechanisms throughout the whole microbial world that actually cope with this problem. And the first one is called the high salt-in mechanism. So these organisms do accumulate salt to the same molar value, but the crucial difference is they switch the cation. So instead of sodium, they have potassium. And again, as Dr. Baker will explain as part of his lectures, potassium is a much friendlier sort of cation than sodium. They still keep chloride as the anion, but they accumulate essentially potassium chloride. So in 4 molar sodium chloride, they will essentially have 4 molar potassium chloride inside the cells. The alternative mechanism is called low salt-in, so as that would suggest they actually keep the salt out of the cell, and they accumulate small molecular weight organic compounds. And these are usually called compatible solutes. You might see them called osmolytes as well, but I'll use the words compatible solutes. And they're basically synthesized de novo, or they can be taken up from the medium, or a precursor can be taken up from the medium and metabolized into the compatible solute, and they replace the sodium chloride. So instead of having the 4 molar sodium chloride, you have an equivalent osmotic potential in an organic compatible solute. In all the undergraduate students I've come across this concept before, though it was quite a while ago, and that was in your, not far away from this time of year in first year when you did first year practical with me with Denaliella. And if you recall back these misty days when you were much younger, you sort of took the Denaliella, put them into different salt concentrations, and they made different amounts of glycerol. So glycerol is a good example of a compatible solute. So if you double the salt, you double the glycerol. So that is the example of a low salt-in mechanism, and I will talk about that one a little bit later. Okay, I'm just looking at the time there. It's not quite up, but actually that's really quite a good place for me to stop. I think rather than try to squeeze in the last little bit, I'll cover that in tomorrow's lecture. So basically I'll carry on with halophiles tomorrow and tell you a bit more about these 2 mechanisms of growth. Okay, thanks very much.